1. Prepare a TS of the given specimen A and identify giving reasons. Draw the ground plan and label the parts. Leave the preparation for valuation. Specimen A would be dicot stem or monocot stem or dicot root or monocot root. Mark is distributed as preparation 2 marks, diagram 1 mark, labeling half mark, at least 2 main parts, identification half marks, 2 marks for reason, in which 2 features for stem or root and other 2 for dicot or monocot is needed. Section of dicot stem. Ground plan of dicot stem. Draw this diagram and at least label any 2 parts. Reason for identification. The material is stem because cuticle is present. 2. Xylem is endark. 3. Vascular bundles are conjoined and collateral. Write any 2. Material is dicot because 1. Vascular bundle open. 2. Vascular bundles limited in number arranged in a broken ring. 3. Pith large. So the given specimen is identified as a dicot stem. Section of monocot stem. Diagrammatic view of monocot stem. Reason. Material is stem because 1. Cuticle is present. 2. Xylem is endark. 3. Vascular bundles are conjoined and collateral. Material is monocot because 1. Vascular bundles numerous scattered in ground tissue. 2. Vascular bundles closed. 3. Pith absent. So the material is identified as monocot stem. Section of monocot root. Diagrammatic view of monocot root. Reason. Material is root because 1. Cuticle absent. 2. Xylem exarch. 3. Root hair present. Material is monocot because xylem and phloem numerous. 2. Xylem vessels are round in shape. 3. Pith is large. So the material is monocot root or the given specimen is a monocot root. Section of dicot root. Diagrammatic view of dicot root. Reason. Material is root because 1. Radial vascular bundle. 2. Exarch xylem. Material is dicot because 2 to 6 xylem and phloem bundles. Polygonal shaped xylem. The given material is identified as dicot root. 2. Identify the material B and C at site by giving reasons. Mark is distributed as name of specimen and its specified part half marks, any one reason for its identification half marks. Identification Oscillatoria filament. Reason filamentous blue-green algae. 2. Cells are broader than long. Identification as mycelium with sporangia. Reasons spores are produced in sporangia. 2. Mycelium is branched and multinucleated. Identified as Spirogyra vegetative filament. Reason cells have ribbon-shaped chloroplast. 2. Filamentous green algae. Identification Moss protonyma. Reason branch thread-like structure grows from spore. Gametophyte of bryophyte. Identification Nephrolipis or fern prothallus. Reason 1. Heart-shaped structure. 2. Gametophyte of pteridophyte. Identification Agaricus basidocarp. Reason umbrella-shaped structure. Basidocarp produce basidospore. Identification Sargassum thallus. Reason erect and branch thallus. 2. Thallus is differentiated into fold-fast, stipe and frond. Identification Funaria gametophyte with sporophyte. Reason Plant body consists of gametophyte and sporophyte. 2. Gametophyte differentiated into root like, stem like, and leaf like parts. Identification Nephrolipis or fern sporophyte. Reason Sporophytic plant body is differentiated into root, stem, and leaves. 2. Spores are released from sporophylls. Identification Pinus male cone. Reason Male reproductive organ which consists of microsporophylls. 2. Smaller than female cone. Identification Pinus female cone. Reason Larger than male cone. 2. Female reproductive organ which consists of megasporophylls. Material C is the photograph of bioreactor or BT cotton or cloning vector. Marks distribution half mark for identification and half marks for reason of identification. Identification BT cotton. Reason it is a genetically modified cotton plant resistant to insects. The soil Bacterium Bacillus thuringiensis produce crystal protein that are toxic to the larvae of insects. Identification Cloning vector. Reason Vehicle which carry foreign gene into host. 2. Contain ORI site, selectable marker and single recognition sequence. Identification Bioreactor. Reason Bioreactor are vessels in which raw materials are biologically converted into specific product in a large scale. 3. Identify the given stage D of mitosis and give reason. Identification half mark, reason half mark. Identification metaphase. Reason 1. Nuclear membrane and nucleolus completely disappear. 2. Chromosomes are arranged in the equatorial plane. Identification anaphase. Reason the spindle fibers gradually shorten and the chromatids separate and move towards their respective poles. 4. Write the aim of the experiment E. Draw and label the parts. Aim half mark, label diagram 1 mark. Paper chromatography. Aim to separate chloroplast pigment from chloroplast extract by paper chromatography. Labeled diagram of paper chromatography. Hydrilla experiment. Aim to demonstrate the evolution of oxygen during photosynthesis. Label diagram of Hydrilla experiment. Viva questions will be related to the physiological experiments. 5. Observe the given flower. F. Describe its floral wall, calyx or corolla or gynoecium. Anyone using 3 technical terms. 3 technical terms, 1.5 marks. Solanaceae. Calyx, sepal 5, gamma sepalus, valvate. Corolla, petal 5, gamma petalis, twisted. Androecium, stamens 5, epipetalous. Gynoecium, bicarpillary, syncarpous, ovary superior. 6. Write down the ecological interaction of the specimen G. Name of interaction 1 marks. Description 1 marks. Name of interaction mutualism. Example lichen. Description lichen consists of an algae and a fungus. The algae produce food. The fungus protect the algae, absorb water and minerals and attach the algae to the substratum. Name of interaction commensalism. Example Vanda. Description Vanda is an epiphyte. The epiphyte is benefited but it does not harm the tree. It gets better sunlight and can absorb water from atmospheric moisture by velamen roots. Name of interaction parasitism. Example Loranthus or Cuscuta. Description Loranthus is a partial stem parasite. The adventitious roots are modified into Hastoria. The Hastoria penetrate into the body of the host and absorb food. 8. Prepare a CS of the given specimen H. Draw diagram and label any two parts. Leave the preparation for valuation. Section 1 marks. Label diagram 2 marks. Label any three parts. Label diagram of TS of anther. 2 marks for practical diary.